In this quick overview, you will be introduced to PCR, or the polymerase chain reaction, and its uses, and you will also learn how this process works to amplify DNA. PCR is used in the lab in order to amplify small amounts of DNA, and it has a variety of uses in science and medicine. This allows scientists to study a specific gene of choice. PCR can also be used to modify gene fragments. It's often used in forensics in order to amplify samples to perform testing, and other fields such as archaeology use this for DNA analysis of specimens. Other uses include using PCR to study evolution and species relatedness, and in medicine in particular, this can allow doctors to detect for the presence of a pathogen in a patient. We've seen this a lot with the COVID-19 PCR test. And importantly, PCR can also allow doctors to screen for inherited disorders, perform paternity testing, and to map hereditary traits. So this technique has a ton of uses, and it's actually really easy to set up and perform once you have all the materials that you need. There are only 5 reagents that are needed for this process. You need a DNA template. This is the gene of interest that you want to study and amplify. You also need primers. These are small pieces of DNA which are required to start the PCR process. You also need an enzyme called DNA polymerase. And this is the enzyme that's actually responsible for reading your DNA template and making new copies of DNA. And DNA polymerase needs dNTPs, or free nucleotides, that it can link together to make those new copies. These 4 reagents are mixed into a test tube containing buffer with cofactors, and that's just responsible for ensuring that all of your reagents will function properly when your reaction is left to run. So how this process actually works, so you will mix all 5 of those reagents into a test tube, and you put it in a machine called a thermocycler. And this machine works by cycling between different temperatures. There are 3 different cycles or stages that the thermocycler will run through. The first is the denaturation phase. During this phase, the reaction is heated to a very high temperature, and that causes your 2 DNA strands of your template to separate. Then is the annealing phase. During this phase, the temperature is actually lowered, and that allows the primers to attach to those individual strands of DNA. And finally, the temperature is changed once more to enable DNA polymerase to attach to the template and to make new copies using those dNTPs. And this is referred to as the extension phase because you're extending and creating your new copy. What's really cool about PCR is, depending on how much DNA you need for whatever test you're going to perform, you can run this through multiple cycles. And every time you run a cycle, not only can your original template be used to make new DNA, but those new copies that you made in the first cycle can also be used. And so you can actually exponentially increase your starting material. So by your nth cycle, you will have 2 to the nth copies. So it's a really quick way to amplify the DNA that you need for your test. This test overall takes between an hour and 2 hours, depending on kind of the length of your original template. And so in summary, PCR is a widely used tool in research, medicine, and forensics. A PCR reaction can be set up in a matter of minutes, and within less than 2 hours, you can amplify a sample of DNA. And you, in this class, will be isolating a gene of choice and using PCR to amplify it in this course. Now you have a quick overview of how that works.